endoplasmic reticulum, and we have smooth endoplasmic reticulum, or SER. And these are going to differ. So our rough endoplasmic reticulum, it's this whole series of membranes here. And we notice that it's got little things stuck to it, right? These little bulges, these little circles. Well, those are actually ribosomes. So our rough endoplasmic reticulum contain ribosomes for protein synthesis. So proteins are made on the ribosomes that are attached to our rough endoplasmic reticulum, and the rough endoplasmic reticulum modifies and packages the proteins that are made on these ribosomes and transports them to other organelles or secretes them from the cell. Now, generally, they have to pass through another organelle called the Golgi apparatus, and we'll talk about that next, before it's secreted from the cell, but more on that soon. Now, our smooth endoplasmic reticulum, if we look at this whole network of membranes, it looks pretty smooth, right? So these lack ribosomes. Instead, they contain enzymes for steroid synthesis and inactivation. So our smooth endoplasmic reticulum is going to produce lipids, steroid hormones. We're going to see it coming up in our endocrine system. It also stores calcium, and this is going to be a key player in our striated muscles and muscle contraction. And our smooth endoplasmic reticulum detoxifies and inactivates various compounds. So we can look at our rough endoplasmic reticulum. Here we see parts of the membrane itself, and we see the ribosome attached to it. And the ribosome is converting messenger RNA into a protein. So it's a protein factory. It's making a protein. And that protein, as it's being made, is budding off into the rough endoplasmic reticulum. And then, from inside the cistern of the rough endoplasmic reticulum, the protein begins to fold into a functional shape. So short sugar chains might be attached to the protein, forming a glycoprotein, and then the protein gets packaged. You see that here. So the protein gets packaged into tiny membranous sacs called transport vesicles. Those transport vesicles are going to bud off, and then it gets sent to the Golgi apparatus for further processing of that protein. So let's look at the Golgi apparatus. So the Golgi apparatus can be likened to the post office. The Golgi apparatus is involved with the packaging of proteins and the distribution, the shipping of those proteins. So a number of things happen at the post office, right? If we've got a package, we need to put in bubble wrap to protect it, or we need to modify the package. Maybe we add some tape to it so that the box doesn't open. So at the Golgi complex, there's modification of proteins and lipids. There's the addition of sugars to form glycoproteins and glycolipids, and the packaging of proteins into more vesicles for distribution either within the cell or to lysosomes or to other organelles or for external secretion. So this electron micrograph here just demonstrates these long cisterns of our Golgi complex. And typically our Golgi complex is located fairly near to our endoplasmic reticulum, and it receives vesicles from the endoplasmic reticulum and will modify the contents of the vesicles, the proteins, and so that modification happens all throughout this post office of the Golgi apparatus, and then it creates a secretory vesicle. And in that secretory vesicle that has the package of our protein within it will decide where to send that protein or other content. Will it send it outside the cell or distribute it to another organelle within the body of that cell? And then finally, we have our nucleus. The cell nucleus is considered the storehouse of genetic information. I like to liken it to the castle of our kingdom, where the king and queen, the keepers of the information, of knowledge, of decisions, are made. So it's the storehouse of our genetic information. Our nucleus has within it, or sort of lining it, I should say, nuclear pores. And these nuclear pores are very restrictive. They're sort of like the moat around the kingdom, with just one gate, one drawbridge that allows transportation in and out, right? But in actuality, our cell nucleus has many of these drawbridges, but still restricting, my apologies there, still restricting the movement of particles in or out of the nucleus, protecting the nucleus and all its genetic information. So generally, things that move into the nucleus include hormones. Also, different pharmaceutical drugs may gain access to the nucleus depending on their structure. And moving out of the nuclear pores tend to be our messenger RNA, which usually goes to the ribosome to make proteins. Now, if we look within the nucleus, we've got our nuclear envelope with the nuclear pores that allow things in and out. We have our chromatin, which is a combination of DNA and proteins, or histones, and we have our nucleolus, which is a dark area that contains the genes that code for our ribosomal RNA. And that brings us to the end of our organelle discussion. Thank you so much.